Greetings, Biology 102. Welcome to your first video, your first class. I'm so excited to start the semester with you. And so, of course, because it's the first day of class or the first week of class, we're going to have a little run through about the syllabus. Now, while we talk about the syllabus, we're also going to move through an exercise, a module, which is going to basically be the format that we're going to do our lectures in. So this is kind of a 2-for-1 situation where you get to learn about the syllabus, and at the same time, you'll kind of get an idea of how we're going to go through our lectures and our classes. So the first part of the syllabus is describing what this class is about. Now remember, this is general biology. We're going to run through all of general biology from things on a molecular level all the way up to ecology, evolution, and biomes, ecosystems, and conservation. And so this is a big class. There's a lot to learn. My priority as a teacher is not just to give you an idea of biology and what that means in your daily life, but also to give you tools and ways to talk about biology to your friends, to your family, to your peers, to other students. It's a little tough in this situation. It's a little hard in this kind of environment. Remember, we're going through COVID and all these things, and we've been put into this remote learning atmosphere, and it's definitely very challenging. But I'm still going to try to give you the tools that you can use to identify your own learning types and to carry those tools on to other classes. Maybe you use them this semester, or maybe those will carry over to semesters later when we're learning in person. And so that's my goal as a teacher. As far as our learning objectives go, one of those learning objectives is learning to speak with vocabulary that is specific to science. So science has a lot of vocabulary that is very, very specific, and that's for good reason. And so we're going to learn what that is. We also want to learn about genetics and how genes work. At the very minimum, we want to know what the outcome of a simple genetic cross would be and how to predict that. On top of that, we'll discuss cellular reproduction as well as the scientific method. We're actually going to start the semester with the scientific method, and we'll be touching upon that concept throughout the semester. And lastly, through our lecture content, I would really like you to learn about the ways that you can gather and interpret primary literature. Right now, you might or might not know what primary literature is, but this is going to be a running theme in this class. I think it's really important to understand what primary literature is, equally important to know how to get it, and also how to evaluate other information using primary literature. Primary literature is kind of the thing that I never knew was what science really is. We learn a lot about science through textbooks and through other information sources that kind of trickle down, but we never really get a clear understanding of how that information is structured in the very beginning. Like, who decides what science and is not science, right? Well, there's a really clear process, and we're going to learn about that process. And once you learn about that process, hopefully you'll gain a value for that, and you'll understand why it's so important to root our understanding of nature in primary literature rather than other people's interpretations and conjecture and all these other wild things that we're exposed to a majority of the time. As far as our laboratory goes, this is going to be an especially challenging task. How are we going to have a laboratory remotely? And so my goal is to create a laboratory that you can do at home. Something that will be interesting, engaging, involve activities, and it's something that you can find resources for in your own home or around your home, in your local environment, basically. So these laboratories are not going to be as broad, and we're not going to do as many activities as a laboratory you would do in person, but we're going to focus on one or two things, but really get into it. And hopefully you will experience a very in-depth understanding of 1, 2, maybe 3 different concepts in every laboratory. That's going to be the compromise for not having a laboratory space, but I think it might actually be better in the end. You might remember these laboratories a little clearer. Overall, my goal in laboratory is to have you learn about experimental design, how to make a good experiment, how to apply the scientific method and make it a process, not just a framework, but actually to apply it to a situation. And also to identify scientific tools and how we use them to measure, engage, and evaluate nature. Lastly, I'd like you to learn how to explore and evaluate nature all around you. And I really do mean all around you. When you look at a piece of bread that has mold on it, I'd like you to understand just what's going on there, the interactions that have created the environment for this fungi to live on bread, what bread is, what kind of molecules make up bread. I'd like you to go outside for a walk and be able to see things like birds and trees and see it through a different lens, the lens of an ecologist. Look at trees across a landscape and wonder about the patterns and try to understand what hydrological cycles might create the kinds of landscapes you see. And I'd like you to have some insight, some depth to at least the questions you're asking about what you're seeing. That's kind of the magic of biology, and hopefully those are practices that you'll take from our laboratories. As you watch our laboratory and lecture videos, you should be taking notes, and I'll actually grade those notes and have you submit them. But the most important reason you're going to be taking those notes is that when you move on to the next question, you're not going to have access to this video, and to answer the question, if you don't know the answer, it would take a while to go back and forth in a 5-minute video to find the right answer. So ideally you have your notes and you can answer the question based on your notes. That's also a really good way to see if you're picking up on the important information that I'm trying to convey. And as we move through, you might get better at identifying what the important concepts are that you should be taking notes for. So as you watch the videos, take notes, and then when you're asked a question in the next part of the module, use those notes to answer the question, and that's how you're going to do your labs and your lectures. So at the very end, I might ask you to take a picture of your notes and upload that. But ideally, through all of the lectures and labs, you should be gathering 2 sets of notes. One of those is for your lectures, and the other is in your lab manual. So move on to the next question. It'll be very easy. You're not going to need notes for this next question. But from here on out, you're going to want to get a pencil and a paper and be taking notes whenever there's video content rolling during a lecture or a lab, including this one.